Okay, everybody at the screen. No, I'm not going to repeat. Now, just briefly, I'm from the University Hospital in Schleswig-Holstein, the north of Germany, and we do translational bioinformatics and medical data science and work a lot with the bioportal. And I've already learned a lot today, so I'm very happy to be here. To have everybody on the same footing, what are molecular tumor boards? Well, molecular tumor boards are at the forefront of precision oncology nowadays. Currently, if you're a cancer patient and you're treated according to guidelines, it depends on the locus of your tumor, be that in breast, colon, or lung, like shown here in the middle, whether you're treated according to certain criteria or guidelines. Now, in the future, hopefully, all these still heterogeneous breast, colon, or lung cancer cohorts will be stratified according to the molecular makeup, saying certain mutations or certain other markers, and that the patients then can be better stratified and receive then a tailor-made therapy like pembrolizumab for MSI instability, or olaparib for BRCA tumors, or BRAF, then you get dabrafenib, for example. And this is something that is already implemented in molecular tumor boards. In molecular tumor boards, people are included that have run out of that are progressing after standard of care, where the doctors don't know what to do with these patients. Also, other patients are included in these molecular tumor boards, which have rare tumors, are resistant to treatment, or are surprise responders. And what's happened in the tumor is that blood and tissue samples are being taken, sequenced, you do bioinformatics analysis and annotation of the variants. We usually do whole exome sequencing of tumor normal pairs, but also whole genome sequencing. And then in an interdisciplinary board consisting of oncologists, pathologists, human geneticists, and bioinformatics, and therapy recommendation is basically discussed for the patient that usually consists of drug repurposing, off-label drug use. We try to include the patient in clinical trials, and also then, of course, try to create research data for future studies. And of course, molecular tumor boards have already proven to show some survival benefit for the patients. Now, where does the bioportal come into play? And for that, a little bit of history. There was a medical informatics initiative that was funded by the German Ministry of Education and Research that ran from 2018 to 22. And the idea was to make patient data in the heterogeneous IT infrastructure of the clinics accessible for research. They want to digitalize medicine and really then found data integration centers. And the development of within these four consortia, HIMED, Smith, DFuture, and Miracum, was basically done in use cases across all of Germany. And one of these use cases was this use case 3, where you want to introduce IT infrastructures and sequencing in molecular tumor boards, from knowledge to joint action between research and patient care. The goal was standardize molecular tumor boards, create common workflows, have an idea on visualization large-scale data, use open standards, open source data, integrate the visualization and the data into local hospitality, and develop study cohort. And what does it scream for? Of course, the solution was to use cBioportal for that and adapt it to the use of molecular tumor board. And at that time, also, there were representatives from this Miracum consortium that developed this software at MSK, got plenty of support, but then we went on basically on our own journey to branch of the development of the cBioportal. And we started basically to design a molecular tumor board as a registry study. And you know these regular screenshots where on top you have then the study of the molecular tumor board. And this is a screenshot of the cBioportal we run now locally in our university hospital, where we have now 1,800 tumor board patients included. And we can now go through individual patients in this list, which you know, I just erased the patient names here, and have the nice annotations due to the cBioportal for their mutations, the history, and the clinical data. Now, what did we do extra? You know, the patient history, you know, the mutations, but we added some extra, and the medical doctors nicely use basically the added annotation linked to all of the OncoKB and other databases and the clinical history. We have this module Patients Like Me, where you can find, just based on the mutations, similar patients. But you can also have this view on the clinical data, and you have some extra tabs where you can add pathology reports, bioinformatics reports, and others. And the central module that we develop is basically a structured therapy recommendation. In this tab, you basically have on the left-hand side, there are free text where the medical doctor can basically do his research, or she can do her research, add the text based on the molecular profile, what should be the therapy recommendation. And then on the right-hand side, you can then have the molecular reasoning, why you give a certain treatment, a certain drug, based on which molecular evidence level, what is the accompanying literature with that. And you can then also prioritize different choices for therapy. And this is then usually pre-filled. And then in the final tumor board session, we just go through the patients one by one, discuss the text, discuss the prioritization, and maybe change this a little bit. But this streamlines the molecular tumor board session very effectively, so that we can also discuss many patients in a very short amount of time and have directly the documentation of the therapy recommendation in cBioPortal, which can be right back into the hospital information system later on. We also added clinical trial search because we want to include patients in clinical trials and can, according to the molecular profile, the place where the patient lives and other parameters, can try and look for an open clinical trial to include the patient. So we also have some other minor optimizations, EMA approval status, Genome Nexus as a database for annotation and such, and it actually works pretty well in our hospital. Now, the big thing is, if you have this loop of patient data, patient samples, the pathology, sequencing, bioinformatics, and molecular tumor board, the hard part is really getting basically patient information and this molecular data together. And because of this difficulty, we not only took cBioPortal by itself, but embedded it into a larger software and toolbox. And particularly, we Dockerized everything, added Keycloak for identity management, and added Fire server to host the diagnostics information together with the molecular tumor, with the cBioPortal. And most importantly, also, we created some tools which we call MTB importer and also then exporter, as you can see along here. And the idea is that we have this heterogeneous data from the clinics. We get CSV files with the patient data. We have PDF reports from pathology from the bioinformatics, and we have the mutational data, VCF or MAF files from the sequencing. And we take all this complex data, and the MTB importer basically creates one cBioPortal study out of that, which we can then readily import into cBioPortal. Additionally, we have the OncoTree terminology service running to annotate the data, have some internal quality control, to have well annotated and well curated data that enters cBioPortal. We also have in the end an exporter where we take the FHIR data from the diagnostics that has been done in the molecular tumor board session, convert it back to CSV because that is what our clinical hospital information system needs, and write then the information from the diagnostics back into the clinical information system. So this way, cBioPortal is fully integrated in our system, and we have then now really closed the loop and fully digitalized due to cBioPortal our molecular tumor board. Question is it really good and efficient? Because the head of the clinics is not interested in the patients, but in the money and how much revenue we can get. And speaking then about another project that is the model project genome sequencing that is currently running in Germany. And there is the idea, funded by the Federal Institute for Drugs and Medical Devices, that you should get precision medicine into the clinics and patient care for rare diseases and oncological diseases. And we will get about 6,500 euros per patient case in oncology, that is for sequencing, pathology, reporting, and everything included. And the requirement that you get the money is also to upload standardized data, about 300 variables per patient, plus the raw data to central genome centers, which are dispersed across Germany, just like the clinics you can see here in green. And because we use the cBioPortal for molecular tumor board, we are really time efficient for MTB preparations and sessions. We have a structured data import and export, and also this documentation personnel we can really save because we do away with, because we do basically the documentation right in cBioPortal. And to do that, we now calculated that we only need 8% of the total cost of the 6,500 euros per patient for bioinformatics, and in the end have a 25% revenue per patient. And this is quite unique in Germany. And basically, this actually proves that efficient IT infrastructures pay off in the long run. What does it now finally have to do to cBioPortal throughout Germany? Well, there is a follow-up consortium on this medical informatics initiative, which is called PM4Onco, that is now trying to basically, as a cross-consortium use case, trying to harmonize molecular tumor boards in Germany throughout, and it also includes not only hospitals, but also cancer registries, or the consortium from the Medical Informatics Initiative, and national research infrastructure. And the idea here is that in 8 work packages, we all have in work package 5 also the visualization analytics and a rollout. And the visual analytics is basically cBioPortal. We're trying to get all of this data into cBioPortal, visualize it, and then also use it later on for research. And we're trying to roll out these cBioPortal ideas then to hospitals in Germany. And in the course of this PM4Onco consortium, we are having current and future developments for the molecular tumor boards. One is, for example, patient-reported outcomes that in the cBioPortal timeline, which is shown here, that we can basically look at certain time points and then see what is the personal status of the patient, how is it improved before and after, and basically can see basically the patient status over time and then align it with treatment. Another idea is to look at patient similarity. Have we found already similar patients to the current patient that is being discussed? And we are developing now measures and metrics for comparing patients according to mutations and clinical data. This is basically just a mock-up we have done so far, but this is under active development. We have further ideas which we want to implement: patient follow-up, pharmacogenomics, monitoring tool for cBioPortal itself, installation support, and also integrating cBioPortal with decision support tool together with the so-called knowledge connector from the German Cancer Research Center. So ideas are there in terms of the molecular tumor boards, and I'm very glad that I've heard a lot more of ideas how include imaging and other data types. So I think the opportunity is out there. And in summary now, what we have done, I think we have proven effectively that cBioPortal is a very good tool for molecular tumor boards as a one-stop shop for many data entities. And we have many ideas for extension, and there is a rollout in Germany going on that we introduce cBioPortal to the different hospitals. But of course, we are only a small development team, and I've seen how much I actually have missed in terms of community efforts, which we also would like to reach out to and basically want to implement. So we really seek realignment with the main cBioPortal development, what's going on in the Netherlands and in the US, and really have a good exchange with a larger community again. All the code base which we have developed is on GitHub, freely available for download and review and critics, of course. And with that, I would like to acknowledge the development people in PM4Onco in Lübeck, in Erlangen and Freiburg, and all the other people that have contributed substantially to this development from this consortium. Thank you very much for your attention.
Thank you very much. Very impressive how far you have penetrated the primary care already. So I was wondering to what extent you are connected to other European initiatives, like, for instance, the Genomic Data Infrastructure Initiative. And we could also talk about Kendall, but we can do that offline because we represent it, of course. But are you connected to GDI?
No, we are not actually. I mean, there were plans also to connect the European health data space and all of these likes, but basically in the daily business and due to the small development teams and the other obstacles we had, this is something we have to like put on the side first. But this is, of course, something we really have to and want to do. Yes.
There are no more questions now. Thank you very much.
Thank you very much.
We will have, I think, a little bit of time at the end of this session for a more general discussion.
Okay. Thanks a lot.
Then I'd like to move on to the second speaker, Melle Sieswerda from the Netherlands Comprehensive Cancer Organization, if I translate that correctly, IKNL. Hello, everyone.
Yeah.
Er gebeurt daar nu wel wat, maar hier niet. Maar klopt het? Misschien dat dat nog gebeurt. Deze. Ja, en multi-monitors staat op automatisch. Dat zou goed moeten gaan. Just presenter wel. Gaat dit goed? Ja, verrek, we zijn er. Ha! Jongens, techniek staat voor niks. Hi everyone, my name is Melle Sieswerda. I work for IKNL, the Netherlands Comprehensive Cancer Organization. And as a first order of business in this presentation, I would like to retitle the presentation from making cancer data accessible through a common oncology data model to a clinical common data model. I feel a little bit like an outsider here today. I think I'm the only one that doesn't use cBio portal. Oh, thank God. There's people shaking their heads. That's good. Okay, Gerrit, I'll go. Still, I hope to provide you with a few interesting insights here today. So I don't think I'll offend anyone here today if I say that in the quest for finding better cancer treatments, care, everything, data is paramount, right? We need data. Data is very, very, very important. And in the past, we may have gotten lucky a few times, right? The discovery of penicillin was sort of an accident. But nowadays, luck doesn't cut it anymore. We need a focused approach. We need to crunch data, look for patterns, look for correlations, associations, causality, even if we can. If not, we'll never be able to figure out how to best treat this disease. And I think in cancer, if you look at healthcare in total, this is especially difficult because healthcare is not a cancer is not a single disease, right? So as doctors, we already thought that treating patients was different and difficult because they were all individuals. This makes it worse. So where do we start when we approach this? Well, I think it makes complete sense to start at the cellular level, right? if you want to understand a disease, you can look at what causes cellular replication, what causes DNA mutations, which changes in DNA lie at the basis of this disease. Which pathways can we identify? These are all very relevant questions, and they will help us to figure out how to best find the pathways that are relevant, how to best identify targets for intervention. Right? But as we researchers sometimes do, when we're very, very focused, we sometimes forget that this cancer develops in a patient. We lose track of the overall picture. So this is where I hope my presence here might make a bit of a break from what we discussed earlier. Because if you look at a patient, suddenly a whole different slew of questions pops up, right? You want to know things like, how was this disease diagnosed? How was it treated? What complaints did it cause? Did the treatment also cause side effects? There's so many other questions that we can answer and that we might be interested in. So I work at the Netherlands Comprehensive Cancer Organization, and our main accomplishment is that we carry the Netherlands Cancer Registry. And this is a very big database that we've collected over 30 years now. I think we're touching on 35, going strong. Over 3 million patients are in there. And we use this data to further research, right? We create data sets that you can request and that you can use for your analysis, that you can link with your other research questions if you have them. We actually publish about this data. And we also actively try to bring this data back to the healthcare process by creating dashboards, by creating regional reports, by basically helping other people do research too. However, and this is, I think, where a bit of where the shoe starts to, that doesn't translate. Right, where we run into a few problems. If you collect data for this many patients, you would love to do that automatically. Now, part of the data that we carry is indeed collected automatically. We have interfaces with Palga. I met a few of them, Palga team earlier today, and they are doing wonderful work. We are very happy that they are there and support us in this effort. They actually have an interface with our organization, but the most data that we carry, the clinical data, is still collected manually. We have 124 FTE, so that translates to roughly 200 people that day in, day out collect data from electronic healthcare records to make sure that we abstract that data into information for research. On a yearly basis, we have about 130,000 new cases. And if you divide that by the number of working days per person, you come to this number, 510 cases per day that we register. This is an insane amount. And while you might think that this is feasible, it currently is feasible. We're also looking at an aging population, increasing incidence, and an increasing data hunger. So this doesn't scale. Now, we've been aware of this for a while now. This doesn't come as a surprise, but it turns out that fixing this is actually fairly difficult. At this point, you might be wondering, okay, but we're here at a cBio portal presentation. How does that fit in? Well, wouldn't it be wonderful if we could integrate those two viewpoints, right? The cellular level on one hand and the clinical data on the other hand. So I think the omics part and the molecular part, you guys really got covered. I'm not going to touch upon that, right? You're better at that than I am. That's fine. For the clinical part, that might be a little underrepresented, and we might help with that. And there you might use data from the Netherlands Cancer Registry, or maybe one of the DICA registries, which are also national registries that collect COLI data, right? That could work. But if you want to copy that data from one system to another, right? You need some way to do that, and you need some way to integrate. So I had a quick look at the APIs from cBio portal, and it turns out that the ingestion part is actually fairly nice. That works. I think we can make that work. But couldn't we make that a little bit easier? And that's where common data models come in. So if I mention a common data model, People usually go to this, right? They say, okay, well, we need to make sure that we're all talking about the same thing. We need to harmonize those data sets, right? That's important. If that's what you guys are thinking, I'm very sorry to say, I'm here to tell you you're wrong. This is not what you should focus on. Did I upset anyone? Shoot. Well, actually not shoot, because I'm happy I didn't offend anyone, because it didn't really mean it. Right? This is important. We need to do this. Don't get me wrong. We need to do this. Actually, we're doing this at IK now. We're currently harmonizing all kinds of data sets to make sure that we can have more data available for research. The reason I'm saying this is because I think that we might also focus on the data a little bit further upstream, right? We might actually focus on the clinical part. And if we do that properly, and I would like to emphasize properly here, everybody benefits. And with everybody benefits, I mean, we as Cancer Registry, it's important because we get easier access to data, which is good. But there might also be data that we don't carry, but that you do want to use in your research. But if it's a common data model that you have at the clinic, then you can just easily get the data out and get it flowing and use it. So I'm here to tell you, people, this is what we should focus on. Don't forget the other part, but let's also make sure that we put some effort into the first part. And why is that difficult? Now, I talked to a few people during coffee breaks, and this is basically what I would like to say about why this is difficult, right? Primary and secondary use of data are fundamentally different things. Primary use, life situation, things develop over time, things change, it's cool, it's okay, we got that. Secondary use, you're missing a lot of context. So to make this a little bit more clear, medicine is messy. How many doctors do we have here, actually? That makes 3. I'm glad we have so many researchers here, right? Because this is important to research. Don't get me wrong. But what researchers often forget is that medicine is messy. When we see a patient for the first time, we don't know what he or she has. We collect this information over time. And our understanding of what's going on can change. Diagnosis may be changed, they may be split, they may be merged, it can be all kinds of different things. Typically, as a doctor, when I'm done treating a patient, I don't go back, right? I don't go back and changing my medical records to make sure that future me or cancer registries know exactly what they're looking at. I don't think we as doctors can be expected to do that either. That's not our job. Our job is treating patients. Yes, we understand research is important and we're willing to help if necessary, but we should be able to do the first part as well. And to give you an example of why this is not always easy or straightforward, if you want to derive data from primary care, you need to be able to know where the data comes from. Sorry, I think I forgot a part, right? Yeah, I forget to mention that we need to focus on data derivation. It's right there. You may have read it, but I think that's an important bridge. Need to derive data from the clinical process, not get doctors to register. So if you want to derive data from the clinical process, you need to know where it comes from. And you also need to realize that not always is the clinical data that you need, or sorry, this data for secondary use that you need registered clinically. Things like an incidence date, which is very important for us as a cancer registry, is nowhere to be found in a medical record. It's a combination of things. It's not the onset date time that a doctor might have noted. It's not the recorded date, right? The date that the diagnosis was first entered into the system. No, for us, it's the first date that a biopsy was taken and the cancer was confirmed. So that's reasoning that we use to determine that date. If that date is not available because no biopsy was taken, we go to the next step. If that information is not available, we go to the next step. There's a whole algorithm here that determines the incidence date, which makes it easy to compare incidence dates, survival times, stuff like that. It's important that we do that consistently. Informational morphology or tumor stage. It might come from a diagnosis, it can happen, but it might also be part of a pathology report, or it might also be part of a tumor board report, or it might also be part of. Getting the data consistently and reliably is difficult. Often it's a combination of sources that helps you put things together. Linking that data can also be very difficult. So if we want to do this properly, we require rules and algorithms, but we also make sure that we focus on data as it's recorded clinically. We should not define indicators. We should not say, I want doctors to record the highest systolic blood pressure after admission to the ICU. That's not important. We do want to know when was the patient admitted to the ICU. We do want to know, how do I find the blood pressure? So the granular items are fine. Derivation of an indicator comes later. That's not a doctor's responsibility. That's our as secondary users. If people are frowning, I'd be happy to have discussions later. What makes this very difficult is that we also need to ensure relationships. If you want to derive data, you need to be able to link it in some way. And that linkage is often not clinically relevant. As a doctor, I don't care whether my diagnosis is linked to an encounter. I'm seeing the patient, I'm writing down diagnosis. If I want to go back in time and find who was responsible, suddenly this becomes important. So we need systems that support this. And maybe we need data platforms that support this, I don't know. So to briefly touch upon how this could work out in the future, there's many initiatives already working on common cancer data models. There's many initiatives working on common data models. There's two, I think, that are very important for us in the Netherlands. The first one is Phoenix, which is an initiative started by HL7 Europe, where we're trying to derive a European cancer common data model. This is a very slow process, as you might expect from a group of over 30 participants that meets once a year. Plugin is going a bit quicker, which is a bit of a strange group of people, but we're bringing together people from all kinds of organizations and try to focus on a data model that does what I hope fixes the issue by using FHIR as a common data model for the clinic. And if we do this properly again, we should have something that's suitable for primary and secondary use. So what should that look like? Well, if we take this one step further, together with HealthRI, we're redefining what a data station is. And this is ultimately sort of what we're hoping for. We're hoping to take clinical data, which is depicted on the left, whether that's Epic or Chipsoft or OpenAir or whatever system, Nexus is still here, I think. We want to transfer that data either directly or through a data platform to something that's a FHIR common data model. I'm very happy I'm not the first one to mention FHIR today. How good? Cheers. I don't know where you are, but if we do that, then we can then translate that FHIR common data model to data that's used for research, like OMOP, which was also mentioned earlier. We can use it for the MEDOC data set that DigiOne is proposing. We can use it for the International Patient Summary, the European Patient Summary. We can also create an export for cBioPortal. Well, that would be nice, wouldn't it? But this is just the first step, because if you then also really turn this into a federated thing, and here I was also very happy to see Vantage 6 mentioned by the Hive, then we suddenly have something that we can really also use to unlock that data and access that data in a scalable and safe way. For those of you who are not as familiar with federated learning, this is roughly what that could look like. So if you have a secure research environment on the right, what you can do is you can send across an algorithm that is executed on the federated data station, and the results of that analysis are returned. If possible, this can be completely de-identified. If necessary, you can still use and share identifiable patient data. So wrapping this up, I'm happy to note that registry data is likely to play nice with cBioPortal. We should do that more often. I also think that a clinical common data model will help to increase the data pool size. And right, this helps paint a holistic picture. I think we should keep that in mind when we do things. And the federated use case, well, we just briefly touched upon that, but I really hope and think that that will unlock new use cases and still help hospitals and other organizations to retain control of their data. I hope I told you something new today. If not, I hope I raised some questions or eyebrows. And I'm open for further discussion, questions, praise, whatever you want to throw at me. It's cool. Achter is dat Stefan, heet die? Je hebt al zoveel vragen gesteld, ik weet je naam.
Very nice talk. I was wondering on the FHIR CDM, so that's the common clinical data model that you proposed. So first, my question, is that already there, defined, or is that more like a concept that you're going to work on and that will take a long time until that common data model is defined? And second, is there already that link to cBioPortal? Is that already implemented? So from the FHIR common data model, can we now directly feed it into cBioPortal?
Interesting. Those are two questions. Let me start with the first one. The first answer is yes, it's under development. We last Friday had a session at the HSF working group meeting to discuss a proposal implementation guide that contains a total of 3 resources. So it's really small, but there is a start. And we're hoping to expand it over the coming year, also in collaboration with HealthRI, into something more substantial. But yes, the first steps are definitely being made there. The second question, is there already a transformation from FHIR common data model to cBioPortal? The answer is no. Sorry, but I'd be happy if you could provide input, because that will also maybe shape the common data model. Right? I want to focus on clinicians, but that doesn't mean there can't be sort of a two-way street.
The common data model will be reported on the 8th of December Oncology Node meeting. If you have time, drop by and see how it's developed. Thanks.
Cool. Okay.
So I hope you remember that complicated slide that I showed in my presentation, which I think I forgot to mention that the actual name, and it is the Blueprint of Digital Business Capabilities of the Health Data Access Body. So I had heard the term data station before. I only learned last week what it is kind of about, and I do think that the concept of a data station actually has a place in that model, and it's actually in the data preparation environment. So I think it could also be useful to see how we can line up there to actually make the world a little bit more tangible for normal human beings.
I think that's a very fair point, right? And I know you and Daniel have been in touch on the subject. The depiction I showed was a bit simpler, I think, than the one that he's using, but the overall idea is the same, I hope.
Yeah. So the point why I'm making it is that the blueprint actually is a kind of European reference standard, right, and that can help us a lot if we all agree on the true north.
Hear, hear.
Well, thank you very much.
Thank you.
Then we move on to the guy who has been asking all those questions, apparently. No. Stefan Klein will introduce us to cBioPortal and to medical imaging radiology. So, Stefan.
Yes, thank you. That was your time to take revenge. And just like the previous speaker, I'm actually not an active cBioPortal user, but so I'm learning a lot today. It's very nice to be here. I'm a researcher at Erasmus MC in Rotterdam, and besides that, I'm 1 day a week employed at HealthRI, working on building infrastructure. Yeah, this is my research group at Erasmus MC. It's a group of technical people, a very international group, and we are embedded really in the Department of Radiology and Nuclear Medicine. developing image analysis methods using a lot of AI. So just since we're in Amsterdam, let's show a picture from Rotterdam. This is this is the Erasmus Center, and we are there on the 25th floor of this tower. It's only 40 minutes by train from here. So we are in the Department of Radiology. So the type of images we mainly focus on are CT scans, MRI scans. So there's a lot of information in the scans that the radiologist can see. But what we try to do is to extract more information out of it using the computer, so using AI. And that can be as simple as just contouring organs or tumors. So you can measure the volume, the size, or shape characteristics, or to create models based on analyzing thousands of images automatically. We also try to create, again, nice visualizations. And more, what we are doing is to, from an image, try to estimate some biological property or immediately create a diagnosis. So, for example, an image of a tumor, we try to estimate what is the type of tumor that we are having here, or do a prediction of the future. Oh yes, yeah. So I wanted to say we are not doing this only for images from radiology, but also actually we also have collaborations with gynecology, so they also create images, echoes, and also from ophthalmology, scans of the retina. And all this research, or the main workhorse, is AI. So I think most of you are familiar with it, AI techniques, and within the more narrow group of machine learning models where the computer learns from examples, and then deep learning as a set of algorithms in there. I'm gonna just give one example of our research to make that a bit more concrete. So this was work by a PhD student, Sebastian van der Voort, together with the neuroradiologist Marion Smits, on the AI in brain MRI analysis for glioma characterization. So glioma is a brain tumor. So usually the flow is the patient gets a scan with MRI. So this images you see on the left, the tumor with different contrast. Then there's an operation, and we get so the tumor is taken out. So it's an invasive procedure, of course, and then based on the tissue, some molecular characterization is done to know what exactly type of glioma it was, from the most aggressive to the less aggressive. So that's based on IDH mutation, 1p90q co-deletion, and also pathological grades. So very short, I'm rushing through this. Our goal was to see, can we not predict this molecular outcomes from the images already, so that we already have them before the surgery. And of course, it's not that we are avoiding a lot of surgeries, because usually people want the tumor out anyway. But you can imagine cases here where maybe a patient is very old and already quite frail, and if we would know in advance that the tumor is one of the most low-grade tumors that has a good prognosis, maybe you would say, okay, let's skip the entire surgery. But also, if you know beforehand it's one of the most aggressive ones, you might change your surgery plan. So we developed a deep learning model for this. That it's a multitask architecture. So we start with the input images, so 4 different types of MRI sequences. We do some pre-processing, and then we train a convolutional neural network that solves multiple tasks as one. So first it identifies the tumor, so automatically places a contour around the tumor in 3D, so we know where it is. And second, it tries to predict these categories of types. So that's the approach. And then, inventing the method was actually the easy part. But then, this is a bit more technical view. But actually, what took time was to collect the data to train and validate this model. So the poor PhD student Sebastian was spending like more than a year to get the data from within our hospital, from public data sets, and all harmonize that and prepare it for the final training and validation. In the end, we got good accuracy, like 85% accuracy, and also just some examples from the automatic segmentations. It was published in Neuro-Oncology. So that was just an example to show what kind of research we do, and to show that it relies on data, and it relies on this automated processing of lots of data, basically. So that comes with some challenges. I think Gerrit already mentioned them in his talk, and I think all of you will resonate with this. So we have challenges in data collection, the anonymization. So that refers to a question we had: how do we anonymize the data? So we have tools to strip off the sensitive elements from the DICOM headers. Cleaning up the data, because typically it's a mess when we get it out of the PACS systems from different hospitals. So we have to rename and structure all the data. We need a place to store it somewhere, to maybe share it with other groups, inspecting it and manual annotation. And then finally, the true processing we do on, it requires a lot of computation time to train these deep learning models. And last but not least, integration. So with the different types of other types of data, basically. So maybe cBioPortal could play a role there. And all preferably in a traceable and reproducible way, so that when one student leaves, the next one can take over easily. So we were lucky to be sponsored by a lot of this national and European infrastructure projects, so that we could work on this infrastructure. So HealthRise is in the Netherlands the main initiative here that we are very active in, and also in European projects. The big project like UCAIM has like more than 90 partners working on infrastructure. In these projects, the main tool that we are kind of propagating, or that we are take as a starting point, is XNOT. So XNOT is a kind of research PACS, you could say, a research database for images. It's a platform for this storage, sharing, and management of medical imaging data. It's open source software, so we didn't develop it ourselves. We take it from the US, and it has quite an active community, and it has proven itself in the field. You can always find some things in software that are not perfect, but this does the job already. It's used across the world. One very simple thing that it does, what makes it so strong, is that it imposes a structure. So it imposes a data model, and that's nothing exotic here. It's just like we have projects. Within projects, we have subjects. Then we have different imaging sessions, and within imaging sessions, we have the different scans that are made there. But that's a structure that anyone would come up with. So now it's at least prescribed with a standard naming. Also, we can control the access for each project, read-write access, and also for different projects. So it's mainly built around the DICOM standard for medical imaging, of course, but it's flexible, so it also supports non-DICOM data. So we can easily also include derived data there that's not yet in DICOM format. So that makes it for researchers quite friendly. And last, it has a website, but also an API. And we actually, in our group, we developed the main Python client that for talking to XNOT in a programmatic way. So we have one, just like the cBioPortal.org. We have our we also have our flagship instance, basically. So that's the xnot.healthri.nl. So that's used both for in national projects and multi-center studies to collect the data, but also in these European projects like UCAIM and UCANimage. It's used as the backbone for storing images and sharing. So it's already up for more than 10 years, and it's hosted by a company with all certifications. We have this help desk, so we have this de-identification pipelines that we can recommend. But as I said, it's been playing a role in large European projects like UCANimage, UCAIM, and EOS for Cancer. So we worked on what about cBioPortal? We worked on the integration with cBioPortal in the EOS for Cancer projects. So also thanks to with the input of Ino there. That's where I met Ino once in a call. And that was a nice collaboration because quite quickly we had something working there. So this is the example that was already shown, I think, by Raymond, where that's from cBio. So in cBio portal, we don't store the images themselves, but we store links to that's pretty simple structure of a URL basically that directly brings you to the right page in Ixnat where the data can be viewed basically. And then did some nice magic to show that in this iframe so that it can be shown in cBio portal. So I think that's a nice integration here where we don't need to copy the data back and we can just use Ixnat for what it's good for, but also have a way to view the data while seeing the other types of data. Another extension that we did was try to make a connection between cBio portal and Galaxy as a way to start image processing flows, because as I think was already said, the cBio portal is actually not focused on the raw data, but it's more aimed at this derived data. So okay, then you need also to have a tool to start this processing to derive the data. So that's what we try to do with Galaxy. I'm not 100% sure yet if this is the way to go, to be honest. It feels like a bit complicated glue between different systems, but it was a nice pilot to see that it's feasible. So also in our own research, we are actually moving more and more beyond just working on images ourselves, because we see the whole field is moving towards this more integrated diagnostics. So I included also 2 slides here from a colleague, Gennady Rostupkin. He actually wanted to go here, but he couldn't make it now. He made his whole research line is called imaging genetics, basically, or computational biology, he calls it, but it's really about combining imaging data and genetics data, and both using as much as possible the raw data and combining it all in a single data-driven framework. So I think for this type of research, also maybe cBio portal could play a role. So this is an example of his work on a deep learning approach for genetic data, so which is interpretable by design. So for predicting a personalized risk score, and also showing which SNPs and genes have contributed to that risk prediction. And then, so the next is, this is then really extending it, so to have both we analyze the imaging data using our deep learning method, we analyze the genetics data, and then combine it all. But then, so this kind of flow, it's for me still a bit hard to see whether that actually fits in the cBio portal way of thinking, because here we start more from the raw data. So that's more like a question I throw here in the group, and I hope someone has a good idea. To end, I have compiled a number of thoughts that came up also in the morning. We'll see in the other talks, some have already been answered. So the first is, to me, it seems really a nice tool, but just to be honest, I think in our imaging field, I don't see it really being used, basically. And of course, I think it's mainly focused on the genetics. So it requires already people that work on the combination. So that's what we're moving to. But so I think we have some work to do there to bring it to more communities. Then a question that I have there is, so what is the vision? Should we really copy all the data into cBio portal, make that the primary storage, or should it become more this linking approach where we leave the data in these different platforms like Ixnat for imaging and imaging derived data, and then only link it basically in cBio portal? That's a question mark I have. Just a very practical question to the developers. Can also a user easily upload data into cBio portal, or do you always need the admin that via the backdoor takes it? Because then, if the last case, I feel this is kind of bottleneck. Because this is for me the strong point of Ixnat. If you have the rights, you can upload your data there. So then every PhD student can work on that. But if it all relies on one or a few stressed admins, well, is not easily stressed, I think. But then there's a bottleneck, maybe. So that's still an open question. That I didn't get an answer in the talks earlier. This was already questioned. So what about the integration with AI development? Can we get out the data? So the answer is clearly yes, there's an API. So that's great news that we can load the data that's in cBioPortal in our Python AI development flows. And actually linking to the previous talk also on this towards this federated data analysis, what I was wondering. So with XNOT, what we're working towards, so we have this our flagship instance basically, but actually we are actively working towards this ecosystem of XNOTs in every UMC or big system where that everyone has their own XNOT. So in that way, we make sure that everyone has harmonized the data in the same way, has stored it according to the same data model. And then I think that's the first ingredient you need to do before you can do any federated learning, that you make sure that the data is standardized at every place. So, but that's only for the imaging data that we deal with XNOT. So it would be great if like cBioPortal could be next to that and serve, take care of the other types of the non-imaging data. And I'm thinking as an imaging researcher, there's imaging data and non-imaging data. So maybe we need another system. This would be kind of a dream that if this would work, and then we can do federated analysis on imaging data and all types of other data. Thank you. Any questions? This.
Thanks for this lovely presentation. I had a very practical question. Does the data that you have on images also include the radiology reports? And do you share this data with other organizations?
Backwards. We share the data with other organizations in various ways. So we made some data sets like public, like with a Creative Commons license, but also more often we now get advice, okay, better make sure first people say, yes, I promise to do nothing and get an account. So, but yes, we share it a lot. And sometimes it needs like a data transfer agreement to share it. But then XNOT is a nice practical way to transfer it to someone else or to give them access so that they can do analysis. The text data is nice. Actually, I had that in this discussion slide. So what about free text data? So in our research, we are doing more and more research with that because we found out indeed the large language models, they are perfect in extracting structured data from the free text, blah, blah. And actually in this kind of AI research, for us, the information in the free text, for example, the pathology report is the kind of the ground truth that we try to predict from the MRI. So we recently submitted a paper on that. So yes, we would be very curious to also to streamline that kind of research, because there, of course, we just started again, inventing the wheel, just storing this free text in whatever format. Well, I think there are actually good systems. One system that I know is a CogStack, and that I know they used it in the UK for already for a longer time for dealing with this text data. So, but maybe you're telling me, don't put it in cBioPortal. Maybe that's the place where you gather this free text data. But do you have a suggestion for that? Okay, great. Yeah, thanks.
Great talk. It's somebody else first. Okay, good. Cool. I was actually curious. Since you're going from the imaging sort of to the genomics, are there sort of any imaging tools where they're trying to layer in genomics data?
A bit this work that I showed from my colleague Gennady Ostupkin. He actually published some studies where the integration is really tight. So not just extracting a few measures from imaging and a few measures from the genetic and then doing some low dimensional correlation, but really correlating every pixel from the brain in the MRI to every single SNP. And then to accelerate that, he produced some tools. One of them is, I remember, called Haze, but I can send some links later if you want. because to accelerate that kind of brute force correlations. So I think that's yeah, more in-depth analysis. Is that what you were looking for? Not quite.
Not quite, but that's actually very interesting. I guess I was sort of thinking more from the visualization aspect of like in cBio Portal, we're trying very hard to sort of include additional imaging features and other things. In XNet, are they trying to do the same for genomics, or is there another tool that tries to do that?
Oh, whether we in XNet try to get genomics data. Yeah, that's it. Well, so what I said, XNet is quite flexible, so it supports other types of data. So there's this also, but there we limit ourselves, to be honest, more to like small amounts of data, some small clinical variables, and we have not tried to draw in complete high-dimensional genomics data there in XNet. So yeah, that's the question. Should you get it into XNet and integrate, or get it into cBio Portal? I would say beforehand. I mean, XNet was not designed for this kind of multi-modal data integration. Well, I think cBio Portal had that in mind, right? So it's maybe more logical to think from there and see how, the imaging specialized tool can be dragged into cBio Portal.
And maybe just to answer one of your questions on the last slide. So what could be useful in cBio Portal? It's all about integrating data. So yes, having a link to an image could be useful, but only probably to a subset of specialists who know how to interpret the scan. Like I don't interact with a pathology image because I don't really know what I'm looking at. But what I would like to see from a pathology image is simplified, abstracted data elements, such as what's the tumor cellularity, what's the percent tumor cells, or is there immune cell infiltration, or anything you could derive from that image. And I think the same is true for radiology image.
Yeah.
You can automatically detect the size of a lesion in a given image and maybe then co-register them over time and tell me how a tumor shrunk over time, shrunk or grew, and maybe do that for all tumors in a sample. Now you give an analyst of genomic data enough data to maybe find correlations between tumors that appear to respond more homogeneously, compare those to those that have a more heterogeneous response, are genomically different, etc.
So basically,
simplifying data.
So we should indeed, we do our job, we process this imaging data, and we should store back the derived results in XNet, and then via this bridge, we can get it into cBio Portal.
We would grab those and store them and maybe visualize them in a timeline.
Yeah, that could be, seems like a nice flow. Thanks.
Stefan, thank you very much.
Thank you.
Then we move on to pathology, I guess, to Wendy de Lang, clinical biologist in molecular pathology and scientist, from the University Medical Center in Utrecht. And you will discuss PALGA and molecular diagnostics.
Somewhat like that, yeah, thanks. Thanks for the opportunity to explain what we're doing currently in the pathology land with cBio Portal. I work at UMC Utrecht, but actually what I'm telling here today is more due to my sort of roles in the Dutch Pathology Society. So I'm here like a spokesperson for the entire pathology community. In 2021, actually, the Healthcare Institute in the Netherlands wrote a report regarding molecular diagnostics. What should we improve? And in their report, they aimed for access for all patients to molecular diagnostics. Every patient should have access to the exact same molecular diagnostics. And also the molecular diagnostics should be performed according to generally accepted guidelines and standards, quality standards. And one of the things that was mentioned in that trajectory was that we came up with a quality standard describing how we should organize the molecular diagnostics in the Netherlands. And one of the things that, or actually two things, were that we sort of described the necessity of a learning healthcare system. I think everyone here knows what the benefit of that is. And for us to be able to get to that self-learning healthcare system, we need to have structured or structurized pathology data. So that was actually something that we aimed for arranging that in the next 3 years. Actually, that started in 24, so I think in next year or so we should be done with this, or should have it completed. So this is actually the structure that we came up with, a very basic scheme. So I think it's quite simple to explain here. So in the pathology lab, we perform a molecular test, and that test has sample IDs, patient numbers, and so on. And also we have molecular data. So our software analysis pipelines come up with the data. We combine the two in an XML file, and that XML file can then be analyzed by molecular biologists. And in the end, we end up with a pathology report containing all the molecular data that has been approved by the molecular biologist. And after that check, and when we have that data, it can, on one hand, be used for the pathology report, so it's sent to the treating physician. But on the other hand, we store the data in the PALGA National Pathology Archive. So actually, we use it for the clinic, and also we store it in our database. It sounds quite simple, actually. However, the infrastructure that we've made in the Netherlands for this approach was made in 2018. And in 2024, we asked the pathology lab, so are you using this infrastructure? And this graph shows, unfortunately, that not all labs are using it at the moment. So we have 18 labs in the Netherlands that perform molecular diagnostics, and only 5 use the PALGA or the PALGA protocol module for reporting sequence analysis data. So that's quite limited, 5 of the 18 labs. And we asked the lab, so why aren't you using it? Because it's useful for getting structured data. And they said, well, implementing this protocol is quite time consuming. We're missing local expertise. We feel that the workflow is not as nice or handy as the one that we currently have in our lab. Link with our lab management system is missing. And also the number of fields that need to be added or data needs to be added is quite large. So it's time consuming to use this protocol. So they said, we're not going to use it. On the other hand, there are a few labs that are using it. So here you see that in 2018, the first lab started using the protocol, and currently 2024, approximately 10,000 reports were added. So currently we have quite a lot of data in the PALGA database, which sounds really promising. So we have a workflow up and running that does work for some labs. Not everyone is very enthusiastic to start using it, but there is an infrastructure there that can be used. And then I thought, okay, we're doing very well here in the Netherlands because we have this protocol, we can use it. Okay, some labs are a bit hesitant starting to use it, but if we would say that we need this, that we really want to have the data structured and stored in the PALGA database, we have an infrastructure that can do that. And then IKNL started with a practice monitor project where they tried to use the pathology data and link it and combine it with the clinical data. And they found out that even though we use a structured way of reporting molecular data, the data pretty much could not be used. And then I thought, okay, we're using a protocol, we have structured data, and still we cannot really answer the clinical questions that we want to answer. So are we giving every patient the diagnostics that they should get based on tumor type, for instance? We could not answer that question based on the data that we collected. And IKNL came up with a couple of recommendations. So they said you need to standardize and structurize the data registration in the PALGA protocol module to guarantee consistency and also completeness of the data. You need to expand the module to make the NGS panels computer interpretable and store the tested biomarkers automatically. And also make very clear agreements on uniform reporting to guarantee standardization. So those were the recommendations of the IKNL. And then we said, okay, so we thought we were doing quite okay. And it seems that we're not doing so well, which was for me at least really disappointing. But it also told us that there's work to do. So the problem at the moment is molecular pathology data cannot be used to improve patient care because the data is not uniform, it's not structured, and it's not easily reusable. So therefore, we set ourselves a couple of aims. And the first is the data needs to be structured. Secondly, it needs to be automatically handled. So no manual steps should be required. Currently, 8 of the 18 labs are using the PALGA protocol module. Of course, it needs to be all labs, so 18 out of 18. If we then have a database with molecular pathology data, the data needs to be able to be used by the different hospitals for quality assurances, but also for research purposes. And finally, we feel that there should be a working group. Of course, there always needs to be a working group, but in this case, a working group that's keep track of the fact how is this protocol working and is it still up to date? Do we need to add certain data points, things like that? And in that working group should be people from the pathology departments, but also from PALGA. And we came up with a working plan. It's a 2-year project that we propose. And in this project, we should start with focus groups where we really need to identify what the data points are that we need to store. We need to have consensus. So every lab needs to agree on what data are we going to collect. Then PALGA will need to build a new and updated protocol module so every lab can start implementing that module to allow identical data storage in all the labs. Once the protocol module is built, it first needs to be tested in a few labs. And we said, okay, we have different lab information systems in the Netherlands. So of every LMS, one lab should test the module. And once those tests are successful, we need to start implementing it nationwide. And ideally, we would like to get this done in 2 years' time. And of course, you need a lot of input. So we have an advisory board with the lab information system providers, but also the Healthcare Institute, IKNL, cBioSymposium, so the healthcare insurance companies, the Patient Federation, but also the pulmonologists and the oncologists. So great plan. If we would be able to do this, then we would end up with an infrastructure where we have the data of all the pathology labs. And after the molecular data has been analyzed by the molecular biologists, the data then is stored in the PALGA database. PALGA is already connected to IKNL and DICA, so they can already share data. But if we would have this, and if we would also be able to link the molecular pathology data to the clinical data, we could add everything in cBioPortal. That's why we're here today, and we would be able to use the data for the learning healthcare system. Also, if we have this structured reporting workflow up and running in the Netherlands, other European countries could adapt that workflow with the SDIP. And also currently there is a project in the Netherlands, that Health Rewired project, that's also looking at clinical data. So I'm really talking only about molecular pathology data, but that's pretty much useless if we don't have the clinical data as well. That's organized in a very structured way. Of course, there are different AI initiatives as well that maybe can help us make sense of pathology reports with a lot of free text information in there as well. So focusing today, of course, on cBioPortal, what have we up and running in pathology at the moment? In my opinion, we can use it once for visualization, but also for consultation. So starting with the second, consultation has already been discussed. You can use cBioPortal very nicely for the molecular tumor boards. That's actually something that we started with in the Netherlands, but in a very, very small way. So in 2018, an MTBC bioportal database was constructed where different labs in the Netherlands could add the variants that were discussed in a molecular tumor board. So not the clinical data is in there, only the variants that were discussed. And we use it in Utrecht to see if we found a variant of which we did not know the clinical consequences. We could check whether or not a different center in the Netherlands found that variant as well. And if so, contact them and ask them, hey, what did you discuss about that patient? How was the patient treated? And can we learn from your experiences? So that's one thing that you can do with the data. Visualization, I think we've seen a lot of beautiful screenshots today showing what you could be able to do with the cBioportal visualization. For instance, we in pathology would be interested to see is the percentage of patients with, for instance, an EGFR mutation in lung cancer in our hospital, is it the percentage that you would expect based on what was published in literature? Are we doing okay with our molecular diagnostic workflow? So currently, PALGA made it possible to get PALGA data in cBioportal. And of course, to get the data in cBioportal, you first need to make sure that you collect the data. And here you see a screenshot of the PALGA protocol module. So that's the module that we use to collect the data. And I don't know whether it's readable, but anyway, up here, it says primary tumor location. That's a field that we need to fill out or can fill out. But above that, you have kind of material. And in this case, it's a lung cancer. So no, it's a tumor in the lung. So we report that it's a lung sample. However, if it's a sample in the lung, it doesn't necessarily mean that it's lung cancer. Of course, in the lung, it's usually the case. But for instance, if you would have a sample in the liver, it could be a tumor from different locations. So meaning you need to think about what kind of data fields are required to be able to answer your clinical questions in the end. So in our hospital, we don't fill out primary tumor location. So we don't have the information what the tumor type was. But we do get the data in cBioportal, which is great. But if you don't know the tumor type, what kind of analysis can you do based on that data? And of course, everyone knows garbage in is garbage out, so you really need to think about what data you need to put in there. And also, some fields are optional, some fields need to be filled out, and you need to have consensus on those fields as well. And you don't want the number of obligatory fields to be too large, because that can be sort of a reason why labs say, yeah, we're not going to fill this out, way too much work for us. So it needs to be as easy as possible. Well, we do collect which test was performed, also which platform was used to do the test on, but we don't always record what genes were analyzed. You can imagine it's quite important to know, of course, in which genes you found a mutation, but it's also important to know which genes were tested but were negative. So that's information that's not currently in there and needs to be there. So that's something we need to fix. This is currently a table that with information that we do import in cBioportal. So all this information is gathered, or is there. So the chromosomal location, the variant classification. But the question is, are all labs filling out all the fields? So that's something we need to make sure that that happens. And this is then the final field of, final overview of the information that we collect in cBioportal based on the PALGA data. So that's, for instance, the age of the patient, the sex of the patient, number of samples of the patient, also the number of mutations, the quality of the DNA, the source of the sample. So in this case, it was formalin-fixed paraffin-embedded tissue, the test that was performed, whether or not the quality was sufficient or not. And this is what I already mentioned, that it's tissue source and not specifically tumor type. So that's something we need to fix. So what are we aiming for, or the roadmap? So first of all, we really want to get real-world data in cBioportal. The data that I showed you is still sort of a setup, scrambled data. And we're currently in the process of getting data from the UMC Utrecht in cBioportal and see if we can make sense of it. So that's the first step. Then we want to check in the real world data what we're still missing and what we do and also do not like of the data. Then the outcome of that should be discussed and also discuss the added benefit of having the data of your own center in cBioPortal. Does every lab in the Netherlands feel that it's an added benefit to have access to this data? That's something we want to discuss nationally. And then the fourth step will be to discuss with Palga in what way the molecular protocol needs to be changed. So we need to have a consensus on what the important information is. And then the protocol module should be adjusted accordingly. So we can indeed collect all that data. And in the end, it could be, maybe I should not say it in a meeting like this, but theoretically, if insufficient labs feel that there's added benefit of using cBioPortal, we should consider stopping this project. I must say that based on what I saw today, especially for the MTB, I think we really need to make this happen, but that's my personal opinion. So we'll see where we'll go. Benefits of having the cBioPortal is share variant classification, share also MTB recommendations, also for practice monitoring, also follow patients in time. Of course, we feel this is a multidisciplinary effort, so it's not only pathology, but we really need the oncologists on board as well, because we need the clinical data to make sense of also the pathology data. And we need to do this nationwide, so all pathology labs will be included in this project. That's it from me. So thanks for your attention.
Yeah, thank you very much for this nice talk and this vision of using cBioPortal for pathology. So I can only stress, yes, directly putting pathology information into cBioPortal, use it for molecular tumor boards, great idea. One question is, if you want to do this extra reporting in cBioPortal, is it something you have to do on top of your regular routine work, or do you see then also efficiency gains in the long run, basically, for doing so?
Yeah, it depends on who you ask, I would say, at the moment. Ideally, we want to make this reporting module so efficient that it will take labs less time to get the workflow going. So that's our aim, to make sure that we have the information from the lab information system and the information from the sequencer, so to say, combine this sort of automatically, and after a couple of checks of a molecular biologist, send it automatically to the report. And if we are able to do that, it will save us time, so it will be more efficient. But yeah, we still have to make this happen.
Okay, thanks.
Thank you, Wendy. Two questions. One is, you showed a slide where you said that you would need to develop a new molecular database. Would that not be redundant with the cBioPortal database?
Yeah, during lunch, actually, we had a discussion about this. So it's, yeah, it depends on who you ask, I would say. The current vision or the current idea is that we first gather the pathology data, and after we've made a decision what we want to report, then we send it both to Palga, and from Palga it can go to cBioPortal. I think also after discussions today, we need to start maybe talking to a couple of cBioPortal people. But anyway, about can you also maybe use cBioPortal as your first step, and from cBioPortal get it into Palga. But currently that's not something where Palga is very enthusiastically about. So I really think we need it. And also, I don't know, so what we do in pathology is we annotate the variants, or we classify them, and I personally don't know how. I know that the information is in cBioPortal, but it's based on databases that you get from the internet. So I don't know it's probably very easy to make, but we also want our own classifications to be in there.
Yeah. No, but I think one point to keep in mind that actually it's much easier said than done to develop a new molecular database, because you refer the story where actually cBioPortal now stands dates back, what is it, 17 years or so.
Yeah, on the other hand, we currently have a molecular PALGA database. So we have something that we can sort of expand and make even better than it already is.
And sorry, one other question, you were also referring to agreeing on basically a molecular common data model, to say so, in the Netherlands. And also there I was wondering, I mentioned this morning the ACR Genie project that actually has brought together data from over 200,000 patients and sequencing samples. I would think that you can get a lot of inspiration there and make your life a lot easier rather than try to reinvent that for ourselves.
No, of course, we need to learn from whatever has already been done, definitely.
Can I maybe add to that? Because I think in multiple presentations, the issue still getting the patient care, the people working in patient care to get their data to structurally put it into some database. And I think we are working on that side. So it's not necessarily discussing what we need to report, but also it's to get all the people in the Netherlands to use this molecular protocol. And for that, you need to have everyone agreeing on what should be mandatory fields that you need to fill in. And the majority of these people are not people that are doing research, but that's people that are doing diagnostics, and they want to keep their diagnostic workflow efficient. And the secondary use is a nice to have, but for them, during their normal working day, they need to have a workable workflow with this molecular protocol. So I think that's mainly where these discussions are about, is what do we minimally need to report that's usable also for secondary use, but also in daily practice.
I think what helps is that also healthcare insurance companies have said, okay, if you want to do molecular diagnostics, great, but you need to collect the data in such a way that it can be reusable. So I think we're really getting there, that we'll get national consensus on how to do that.
But also when you think in terms of procedures and logistics, I think also there you can learn a lot from a project that has been able to collect so many molecular data sets from so many different institutes. So also I think looking behind the screens of what ACR Genie has been doing could be quite useful.
See how they do that. Yeah, definitely. Okay.
Really? Thank you very much. Then I'd like to move on to Joep Licht from the data lead from the Hartwig Medical Foundation, correct?
Thank you.
So we are moving to big gene sequencing data.
Chunky data, as we call it.
Chunky.
Yes.
Okay. All right. Let's see.
I was asked to talk a little bit about how we look at cBioPortal, because we don't have a cBioPortal instance of the Hartwig data. And that's not because we don't like cBioPortal, but we think we can get more out of the data if we think about the data in a different way. So hearing some of the talks just before, I think cBioPortal could reposition itself as an analytics platform, not as a genomics platform, where you bring in other pieces of data and you can analyze that data. And why I think that's useful for us is that this is what we want to happen in the Netherlands. We want to be able to learn from the data that is generated in care and make it available for research, and do that in such a way that you can reason about what biomarkers are indicative for response, but also non-response, that you can look at how is your hospital compared to other hospitals. Those types of queries, that's what we want to enable. And we believe that whole genome sequencing is quite rich data. And this is the tool set that we've built to try and squeeze every single last drop out of that is possible. So there's viral integrations, there is immune scoring, there is a whole bunch there, also RNA data. But this is what we think and please tell us if we need to add more. But what would result into a comprehensive view of what the patient is, so that you know if there's a mutation in BRCA, if that was a germline with a coupled LOH, or if that's completely somatically acquired. It has a different indication for the resistance likelihood. So when we looked at cBioPortal, because we want to make our data as freely available as possible, and people kept saying you should put it in cBioPortal, so we took a good look. And there's a lot of things to love. There is this interface that is very intuitive once you know your way around it. But there's a community around it that is developing new ways of visualizations, like with the new structural variations. So that's all very great. There's APIs, there's R and Python bindings, so people can work with this in their favorite way. Another take, and they have integrations with OpenTargets and OncoCKB. Again, so those are all the pluses, but it was built from a gene-centric point of view. Like when this was built, we had panels tumor only, and that was about it. Now we have whole genomes with normals attached to it, where we have things like chromosome arm level events that we now know about and can reason about that are not feasible, or at least from what we saw, to just plug into the model as it is. And it has a variant type focus where we try to take a slightly more holistic view. Like if you're talking about HRD, can you then combine all of those HRD genes into an HR score? Those types of concepts from a whole genome perspective are quite important to us. And of course, with whole genome comes things like non-coding GWAS analysis and those types of things that people want to do on our data that are not necessarily feasible within cBioPortal. And then the RNA is sort of the last thing that we're adding. And I'm not saying that cBioPortal can't be those things. I'm just wondering at how it will become that. I'll come back to that at the end. So this is a study that was done here at the NKI, which we thought was kind of cool, where people looked at non-response biomarkers. So there was this indication if there's a high molecular tumor load with a lot of somatic mutations, you're more likely to respond to immunotherapy. But then, of course, people said, well, that's great. It's a good predictor for response. There's 60%, which most oncologists will be quite happy with as an indication of your patient responding. But in non-response, there's still 20% of people not responding in the non-response group, which means that a doctor is not going to say, I'm not going to give you immunotherapy, because there's still a 20% chance that you're going to respond, which is not that far off from 30%, which is average for a lot of medications. But the team here then looked at, can we purify this marker? So they found 3 additional genes that if you look at those genes and combine them with this TMB, you can get to a group that has 0% response in that group. So this is where you could say, like, well, clinically, we can now not give this drug because the patient will suffer all of the side effects, but none of the benefits. They replicated that in another study. There was only 1 patient there that had a clinical benefit within that group for that biomarker. So they reasoned that this could be a viable thing. Now, what we wanted to be able to do with the data that we get from all the hospitals in the Netherlands is to do this systematically for all biomarkers. So we asked Joseph Usset, who's over in Barcelona, who's done a lot of work on the data sets from Hartwig, on prognostic markers, to do a systematic screen for non-response. And this is just the top 10 of things that he found. So there's much more. There's chromosome level arm events that we don't know how they work, that predict immune chemotherapy response. But these are the ones that we think we can make some sense of. And these are the types of things that I think we need to be able to enable in platforms like cBioPortal or others if we want to drive a learning care system. Because the one that I've highlighted here is in melanomas. And it's a biological mechanism. It's mostly driven by B2M deletions or deactivation. That is, it makes sense. It's involved in the immune response. And if you lose that gene, there's no immune response whatsoever. Now, this is the Kaplan-Meier that we got out of our data on the left, and on the right is as far as I've currently gotten with cBioPortal, and I hope so somewhere after this talk someone can explain me how I can do better. But what you see on the left is all patients on immune therapy stratified by their immune escape status. So we combined a couple of features that are all related to the immune system. 10 out of them are B2M related. One of them is slightly different. And none of them show any durable clinical benefit under immune therapy. While the people that do not have that biomarker, there is some benefit to that treatment. On the right-hand side is what I got out of cBioPortal, and this is all the cases that have a B2M alteration. And I haven't quite been able to make that a specific alteration. I know you probably can, because if you do look at the data, there is the deep deletion here on the right where there's very few positive indicators of response, and most of them are progressive disease or no response whatsoever. So it's in there, I just can't get it out. But these are the types of things in terms of not just one gene being altered, but there's particular alterations within that gene that drive this biomarker that you want to be able to validate and use this platform to do so. And the other thing is that these data sets that we have and also that are in cBioPortal, they might look big in terms of numbers, but once you drill down to a particular cancer type with a particular biomarker, with a particular treatment, numbers get scarily small very quickly. And that is why I would love to see across the Netherlands panel data also coming together. And I think that's what cBioPortal is beautifully built for and could be a really good source where all the data comes together, so that we can go from where we currently are, which is 166 patients. If we want to make that marker that I just showed you clinically actionable, we would need to at least quadruple the size of the data set to be able to get to less than 5% response. And if you want to get to less than 2% response, we need to be 10 times as large. So that's what I call big data. A genome is chunky or fat data. This is the big data that we need to make this a reality. Now, if I look at the international spectrum, most people are now converging on two ways of having data. There's the genetic data, which they're looking at the Global Alliance for Genomics and Health. They have developed a beacon model, and this is a very narrow standard for sharing genomic data. And on the other hand, they have clinical data. And they bring these two together in some ways or forms. This is one example from the data space in the UK, where they have a very large team working on supporting researchers, pulling data from different sources, so from imaging, from genomics, from the clinical data, and then being able to analyze that. And they've made the Relay, it's a piece of open source software, but that gets data from different resources. So we just heard a little bit from imaging. They have a perfectly fine standard by the sounds of it. And they're working on making sure that that all gets shared. So how do you then bring those different data sources that all have their ever so slightly different standards together in a way that works? Now, to me, we need a standard, just like we have for the internet. We have the IP protocol, and we have HTTPS, we have FTP, we have all these things on top of it. We need a standard for genomics, and that was what, to me, should feed into cBioPortal. So if that's Beacon or something else, when you choose something, then you can do the analytics, and you can start to think about federated analytics. Because all of these parties, they tell us, we're never going to put our data out there in a public environment. This needs to be controlled. This needs to stay locally. So you need to think about how to federate these analyses and how your platforms can interact with those federated analyses, because that is going to be, to me, the big unmet need. So let's assume EHDS is the case. All the hospitals have their little clinical data sets sitting there. Some of the big genomics initiatives have their genomics data sets sitting there. Hospitals might have their own. How do you bring that data together in a way that people can actually reason about it, analyze it, and work with it? Now, I don't have all the answers, but we have some things that we have built thus far that might or might not be of interest to you. So these are what we call vignettes, and it's basically our cohort level overview of all the things you can get out of whole genome sequencing. So there's genome-wide features like the genome level of LOH that you have, immune escape mechanisms that you might have, and all the other bits and pieces. And we've done them for about 70 different cancer types for which we feel that we have sufficient data. if you're interested in them, go look at them. They're open CC BY license. You can download them. We update them about 2 times a year to make sure that it stays up to date. And that's sort of a way where we think we can start to think about leveraging the richness of whole genome sequencing data. If people are working with the Hartwig data, please know that there is such a thing as the driver catalog, which does bring things back to a gene level, but then with information about whether that gene is biallelically inactivated or not, and if it's likely the driver of the cancer. So these are all things that are pre-computed in that very complex diagram that I was showing you. And we think that there is a future ahead for these types of solutions. So if we can get to a point where imaging has their standard, genomics has their standard, clinics has a standard, probably FHIR related, we can then build something on top that can reason about those things. And I mean you guys. And we would love to help. Because we need something that has multi-omics, multimodal. We need this to be aggregated across hospitals, across nations, if we want to get the numbers that we need for clinical validity and clinical implementation. These are some of the parties that we are working with to try and make that happen in terms of harmonizing the way we analyze our data and harmonizing how we expose that data. And the last thing is that we're not in this alone. I'm very happy to be here today and meet some like-minded people, but we need you along the way.
Great presentation. Thank you. One question, like I think we're struggling a little bit with fitting too much into cBioPortal. Like, and you sort of just suggested we should try. I think non-coding is really, really complicated. Germline is probably not feasible for cBioPortal because most of our users don't fill out an application, get institutional approval, et cetera, et cetera. So we probably have to limit it to somatic, but that way we reach 100 to 1,000-fold more users. Right? But for your purpose, for example, would it be, you don't always need to squeeze everything into one platform. Would it be possible to have users have access to different platforms that all under the hood reach the same data, and you might just be limited to what you can do with that?
Yeah, I fully agree. Like, I'm not advocating to put everything in there. I'm just saying that there will be a need for such a platform that can combine those different modalities. And yes, as you say, like there is a place where if you've done a genome and you want to just expose those genes that are commonly in the panels and put them in, that you want to do that. But then I do think that there needs to be things in the data model like not measured for all the other experiments so that you can compare panel and genome that has been run with a normal in a sensible manner. But you could make a loader that either talks to a beacon interface or runs on your data that then loads those specific items into cBioPortal that comes with some of the information about the privacy, sensitivity, or not of that data. Because that's still something that we see a lot of hospitals struggling with, is like they want to share their data, but they're wondering like, but how can we share the right parts in the right way?
So when I look at the vignettes, then it looks like, okay, you're getting a window through what is stored in the Hartwig medical database with a visual layer. So you know about research linking in cBioPortal, plug and play, you get a view window. That's also what we saw earlier today during the XNAT presentation. It's just a view window. So then ideally, if a sample was sequenced that's stored at Hartwig, you would want to do resource linking. If you're a molecular biologist, you're happy, you scroll through the images, you get your nice panel data of your local hospital in cBioPortal itself. Hartwig Medical Foundation data, you just see it. If you want to do technical, complicated matters, you're a bioinformatician, and then you need to get the raw data, which is not stored in cBioPortal anyways. So then either you would go to a Hartwig Medical Foundation GCP, or you would take the data to your HPC of your choice. So that could also be an option.
Yep, no, that's definitely an option. But we would rather support one option well than a whole bunch of different ones.
Thank you, Joep. Impressive presentation. If I'm correct, I think I heard you saying that it would not be possible to bring genomics data together at one place in a country.
I don't think that's the way of traveling. I would love it to be just a national data, like other countries have done it.
Yeah,
But within the Netherlands, I see a lot of resistance.
Okay, so then let's try to dissect it. So
Sure.
then it's not the point that it would not legally be possible, but there is some kind of human behavior involved that is kind of impeding this. Is that?
At least that's my personal opinion.
Yeah. Okay. Because I do think that there would be an opportunity combined with what we heard before from Wendy, and also with the upcoming European Health Data Space Regulation. Actually, PALGA could simply apply for a data application for all molecular data, and if that is granted, then simply all of the hospitals need to hand over that in some kind of secure processing environment that you could think of how to organize that. But at least that's our view at HealthRight, that we should not have local secure processing environments, but rather have it as a national shared service. Then we are at the goal that you're aiming for, isn't it?
Yeah, no, I would love to see that happen.
Yeah.
And I think it is definitely technically and legally possible.
Yeah, so we should simply kick some ass and make sure that it's going to happen.
I'm all for it.
Thank you for the great talk. I was curious also with, it seems like there's a lot of, institutions that are maybe not willing to share the data. So one thought is that, patients, they have access to more of the data themselves, maybe. Like they can get it from different types of institutions and piece it together. Do you have any insights or thoughts on like can they, for instance, obtain their own Hardwick Medical Foundation data? And is that another way that we could, directly work with patients, and maybe they are eager to share their, data in some other way if it's sort of if the issues are at the institutional level?
Yeah, I would love for a patient that has received genomics in any hospital to make that request. If I would be a patient somewhere, I would definitely do so. So lo and behold, the hospital ever has to treat me. But I don't know if a hospital would be able to provide access to that data. Like in the US, this happens a lot. A lot of initiatives there that have aggregated data have gone through the consumer, because direct-to-consumer testing is much more of a thing. Here in the Netherlands, we're slightly more paternalistic and protecting people from themselves and from their data. So I would love to see the experiment done to see what happens. Also, like, as it was just mentioned, like under EHDS, like some of these things then become obligatory by law. I'm interested to see what's happening. We want to approach this from what is already possible now. So if there are initiatives that are bringing together these data, like Rewire that was shown in the previous presentation, we're there to make that happen. And if there's a good way to expose a view in cBioPortal to people that already have access to Hardwick data, let's go for it. But yeah, it is something that we're noticing that is difficult, and that's legally difficult for them because it's not been done before, so they're not quite sure what they can or cannot do. It's technically difficult because they're not necessarily used to exposing genomics data in such a way. So there's many factors at play here.
Maybe one more follow-up on that, on the technically possible, legally possible, but maybe not politically. I always wonder for whole genome sequencing data, if you could just limit it to, and you have a matched normal, somatic coding variants, non-synonymous, why couldn't those be made available to the public in a public cBioPortal without a user access sort of agreement, etc.? Just like Peacock has made that same subset of variant.
I think it's possible, but okay, no one has done it.
Okay, the answer, well, here's then the public challenge. Like we've tried to get that data from Hardwick to put into cBioPortal, and we were told that that's not legally possible.
Yeah, but that was prior to the recent European regulation ruling that was in the court about can be considered anonymous. So we should definitely revisit that.
Okay, cool. Okay. Encouragement to get started with this.
Thank you.
We had a bit of time for discussion blocked here. I think we already took quite some time for discussions in connection to the presentations, which was nice, but maybe there's still some topics we would like to discuss in general. For the speakers, for instance, you have thought about wish lists for cBioPortal, and Stefan put them on paper. How you may have thought about them? Have they all been addressed? Are there still some items on your wish list, or for Wendy or others? Nikki, is there anything you think like work to do, inspired by some of the things that passed by and that may trigger your thoughts on?
I think like we've heard about lots of things that are on people's wish lists. Like I heard plugins make it easier to integrate features. I heard data upload. Ironically, I think data upload was an item in our first iteration of the cBioPortal grant from 2017. We put it into the second one again, and we haven't worked on it because it's just, yeah, complex. And refactoring was important during the first grant, so that's what our focus went on. And now refactoring the database is in the second one, so we're just not getting there. So we don't have the bandwidth to do all the amazing things we could do, unfortunately. I think we're also trying to integrate all the up and coming features. Like digital pathology is now pretty much commonplace, so we're trying to integrate that, and you'll hear about that later. I think radiology is still a little further away and maybe an order of magnitude more complex. And then spatial transcriptomics, all those things we can link to, but we're not going to be able to derive data from. And then the whole genome maybe it's not quite in the clinical space. So for example, we just at MSK decided that we're going to switch from our 600, 700x deep targeted panels to the same depth in an exome. Because we realized with increased capacity in sequencing, just switching to like different flow cells, we can, for the same kind of manpower, handling cost is going to be the same, chemistry cost is going to be roughly the same. The only increase is the cost in informatics, storage, and running the pipelines. But we couldn't do the same for a genome yet. And we need the depth. We need the 600, 700x depth in clinical practice. So I'm curious about what other people think when we can get to clinical genomes. Do we have more time to, because otherwise we need to increase or improve our tools for that as well. Or is it going to be an interplay where we keep doing genomes at a smaller scale, like Hardwick, Genomics England, and then we learn from that and add to our panels or exomes the regions that we need to cover. Maybe that's another topic for discussion.
So going to a little different topic, like way in the morning, we saw this slide on the EHDS Digital Business Capabilities Blueprint, really colorful slide. And this afternoon, we've heard a lot of talks about, hey, from Polga, we have this idea to put our data upon request of a data user into a space that someone who wants to use the data can actually use the data. So that's being worked on. From IKNL, it's like, yeah, we are working on it, but hospitals, get your data in order. And then for radiology, XNet is a way to share the data. Stefan, you also indicate that when someone knocks on the door, like, I want to do radiomics data, that you offer a secure processing environment to hand over the data. So what I'm wondering, in the Netherlands, we need to create this blueprint, this infrastructure. Who is coordinating that? So how do we avoid that some of the country go left, depending if you're under the rivers or above the rivers, and that other people are going right?
It's a tough question on the 11th of the 11th.
Isn't that something like Candle, Health Right? But how would Health Right do that?
That's not a challenging question at all. Well, we have a magic solution. So the oncology node, the national oncology node, that's really a collaborative platform where people like you, like us, sit there, really talk about what are the users' needs and the developers' needs, and how to align that with EHDS. There was a question I remember to one of the speaker. Are you ready for the implementation of EHDS? How to incorporate cBio portal into the system? Willing or not, it will be a must. But HealthRI or oncology node, national oncology node or candle could help and support that what we can offer. But from today, I really learned that coming together is really useful, and the users need. And sometimes in the tea break, I heard, I didn't know there is another function. It's great to see. You can use cBio portal in this way and that way. I think this gathering is quite useful for me, at least.
Yeah, let me try to get back to this terrible presentation that I did in the morning on European Health Data Space Regulation. So point is that this European Health Data Space Regulation needs to be implemented in the Netherlands in 3.5 years. And actually, the ministry also doesn't know what they need to do about it. They just know that they need to kind of install 2 authorities, one for primary care, which is the Health Data Authority, and the other is for secondary care, which is the Health Data Access Body that I mentioned. And they undoubtedly will do so because they're obliged to do so. But what happens next and what these authorities are going to do is something that actually we should fuel as communities of professionals, researchers, and experts. And I think that is a big opportunity that with this domain node oncology that Li Fang was referring to, actually should grab and use to the max for all of the things that we have been discussing here. So I really consider this implementation of the EHDS as a once in a lifetime opportunity to really get a lot of our wish lists that we have been discussing here to get them adopted. So yes, it's bureaucratic. Yes, it's fuzzy, but don't forget, it's really an opportunity. And as much, the more that we as community actually get together and define our specs and make that clear to the ministry, the bigger the chances are that we're going to succeed in this.
Just a little ads. When we talk to some European project and their leaders, they are fully aware that the blueprint is there, but we also need to make reality check. Therefore, we are invited to those EU projects to really try in the research project where it goes, where it doesn't. And the Commission also are very clear. EHDS should be a facilitator for research community. The community is not really an intended barrier. So we also need to help the Commission and the ministries to understand where the community can benefit from the framework. So it's really a two-way road. It's not one top-down approach. Although it's a regulation, we have to do it, but we need to tell them how to do it and for the benefit of the research community.
But I still think we're overseeing this. There's this big gap because we're seeing that in diagnostics, we want all the data structurized, but it's not being saved in a structured manner at the moment. So we keep saying, put it in a structured manner, put it in a structured manner, but it's not happening at all the hospitals. We're saying, why are the hospitals not sharing their data? It's because they don't have it in a structurized manner. There are 124 people, I learned today, at the cancer registry making data structurized. So I see these wonderful things that you can do with the data once it's structured and in research. But how do we overcome this gap from this data we have in reports being used by doctors for patients that don't really have time to think of these bioinformatic processes? How do we overcome this gap with all these researchers that can do wonderful things with these data? I think that's still a large problem at the moment.
I'm not sure whether I'm the right one to comment on it at all, but you can discuss this topic for centuries. You can also say, here's the solution, work to this solution. And I think for me, from someone not involved in the detailed discussions, and that's why I know that this is not a very nuanced remark, but it is an approach. It is an approach to say, this is where we work to, and get the data done in a way that they can enter, well, and then look around. We had our experience in the past with one tool that failed. We have seen cBioPortal grow, flourish. You see the community spontaneously grow. That's very strong. So if this is a tool, as a tool, it's not relevant. You need standard data formats. Those data formats you can use in 100 tools. But vice versa, by positioning the tool as something that is very helpful to clinicians as well as researchers, you have a common goal to work to, and it is a different starting point of the discussion.
If I may follow up on your question, my impression is that data that is most easily standardized is data that is coming off a machine, like for instance, imaging data. We've seen that. And data that actually comes from a narrative that doctors are producing, that is the hardest. And of course, as pathologists, we are the exception because we have APALGA protocols, but still. And my impression always was that actually the same would go for genomics data because they also come running off a machine. And in some ways, to my understanding, the workflows behind ACR Genie are also kind of rather automated. So I was wondering why that then not could work for clinical genomics data, and perhaps a bit in a way like when you were saying to perhaps first bring it into cBioPortal and then from there see how you want to kind of what's the term to limit the elements that you really want to have in a pathology report, but then at least you have the whole data set already in a structured way. And if that can work for ACR Genie with 20 institutions from 6 countries, I'm wondering why it could not work for, 18 labs in the Netherlands.
That's a good thought for over the break, I guess. I would like to ask you a last round of applause for the presenters of this session.